and welcome to Conroy Academy. Today we're going to be discussing RNA transcription translation and more importantly gene expression. So right now we have a DNA molecule. This DNA molecule has two strands as you see in our paired up G, C, and T and A. Now we have decided that this molecule, this gene right here needs to be expressed or needs to be actually telling the cell what to do. So pretend like this is your insulin gene. You just ate a whole bunch of sugar and this needs to say, okay, let's make insulin. Now because the DNA does not leave the nucleus, you need something to actually leave the nucleus and go tell the rest of your cell over here what to do. So this is the process known as transcription in making RNA. So the DNA knows that it's supposed to do something. So what happens is your RNA polymerase comes in and attaches here. And it attaches and starts breaking these bonds. And as it's breaking these bonds, it's creating a new strand of RNA that is coded for by these bases. Now, if you remember from previous videos, RNA has very similar base pairing rules, except for instead of having this be paired with a T, you have a U. So out of this DNA, if this strand of DNA over here is known as your template strand, you will produce an RNA that has a U, an A, a C, and a G. Now it's very important that you always transcribe the same strand of DNA known as the template strand, because if you coded for the other one, you would get a different mRNA strand, and that would code for a completely different protein. So this is transcribed. Then, if this is the nuclear membrane, this strand of RNA leaves and goes over to a ribosome. And the ribosome over here has two units. And if you've ever seen a ticker tape back from physics, this runs like a ticker tape through your ribosome, creating your protein. So this goes through and you end up with your protein coming out like so. So this goes through and is read and makes your amino acid sequence here. So this is your protein. Then your protein goes, and it's in this circumstance insulin, goes and into your bloodstream. So this is your bloodstream over here. It goes in and binds with these sugars, brings the sugars back into your cells, and the sugar is broken down by your mitochondria. Then, as soon as you don't need any more insulin, this whole process gets shut down and this gets turned off. So this is only being done when insulin is needed. So it makes it a lot easier to regulate, because you don't want to be making insulin if there's no sugar here for it to get. So as soon as there's too much insulin in your cell, it signals this to be like, oh wait, we don't want any more of this. Stop this process so that we don't have too much insulin, because we don't need insulin. So that's the basics of transcription and translation, which is the DNA going to mRNA, going into your ribosome, and being made into your protein. Now, I mentioned the fact that if there's too much insulin, this stops. That is known as regulation, and when this actually gets turned on, it's known as expression. And you can either activate or repress a gene. So activation is when this is actually turned on. Now, how that's actually done is through feedback mechanisms and this protein here that it makes. So activation, let me go down here. Activation is controlled by, is when your gene actually gets turned on and proteins are made. repression is turned off, equaling no protein. So this is the crux of gene expression. And it's what actually makes sure that you're only making this lovely protein over here, insulin, when you have sugar in your blood. Because without the sugar, the insulin is useless and it just sits there, waste of energy, and you never want to waste energy. So this is just the whole review and big picture of how RNA is made from DNA and then made into your ultimate protein over here. And how it's regulated is actually self-regulation. Now in different cells, different types of cells, there are slight differences, and what actually controls this whole thing is slightly different. Versus prokaryotic versus eukaryotic, something we will be getting into in another video, but it's all about whether activation or repression is occurring. So that is the big picture and all I'm going to do today.